So I'm here today to talk about decoding diversity. Similar to the last talk, what we've done to use HiFi sequencing to look at variation associated with health and disease, but I'm going to come at it from a slightly different angle. So what I want to start with is introducing the PacBio customer base to the Clareo Biosciences family. So we recently opened Clareo Biosciences. We launched in 2024, but it is a company completely enabled and built on HiFi. And what I want to tell you today is the story of how Clareo came to be, why we couldn't have done this with a technology that was not HiFi, and then what we can offer to complement those users that use PacBio technology. So we've developed a suite of custom long-read sequencing assays specifically focused at profiling immune receptors. And what we aim to do with that is answer questions that go from the basic science of how does disease pathogenesis work, how do host pathogens interact in a system, to discovery of biomarkers, companion diagnostics, drug discovery, et cetera. We believe, and I hope to convince you by the end of this talk, that our tools will enable people to discover immune genes associated with response and disease progression, leverage that screening tool to identify people that may respond or may not respond, inclusion, exclusion criteria in clinical trials, and then of course the moonshot hope, right, is to feed that information back to drug companies when they're developing their next generation therapies to make them more broadly reactive for all. So the example I want to use today, there's two examples. I want to talk about vaccine responses and autoimmunity. Let's start with vaccines. So all of us in this room, especially in the last 5 years, know that some vaccines are incredibly robust, but they're not 100% effective, right? They range from sort of an abysmal rate for seasonal flu to quite high vaccine rates with the most recent SARS-CoV-2 vaccine rates. But understanding the factor of why things are differentially responsive are really limited and have been thus far very focused on the vaccine itself. What antigen did you choose? What adjuvant are you using? With very little consideration of the host. And this has led to the idea and the hope that we can develop universal vaccines, one shot that fixes all for the rest of life. So a great example of this is the measles, mumps, and rubella, also particularly timely in today's world. 90% of children in the US get the MMR vaccine, so that's 3 million kids a year, but up to 10% of those children will not respond and remain susceptible to what was the most deadly infectious viral infection until SARS-CoV-2 came around. But only 20% of that variation is understood. And as a parent, this is somewhat terrifying because a small fraction of measles-infected individuals will go on to develop a deadly encephalitis. So our academic group at the University of Louisville has produced new evidence in the last few years showing that the genetic background of the host is critical for understanding why people respond or don't respond in vaccines, and that was completely enabled and only enabled by HiFi data. So we're going to talk a little bit about antibodies. For those of you who haven't had immunology since college, I want to do a quick reminder just so you understand why HiFi data was so critical for this approach. So for those of you that remember, right, your antibody is your canonical Y molecule. The business end of the molecule are these arms, right? They bind the virus, they bind the bacteria, but the action end is the tail. But when you're doing immune profiling methods using NGS or any of the common strategies, you're really only profiling the variation that's in that red business end that binds the specificity of the antibody for the molecule. But you're not looking at the genetic variation in the tail that actually dictates effector function. So we're going to address that in a minute. Additionally, these are loci that were not represented in the era of GWAS. So when we think about HLA and other immune genes that have been associated with disease progression, genes associated with antibody or Ig, immunoglobulin, or T-cell receptors weren't really included. So this is the human genome. This is the oligo coverage for SNPs using the Illumina immune array. You can see here that there's over 9,000 SNPs represented in HLA. We all know that infectious disease associations with HLA are quite common. Even in the NK cell receptors, the KIR receptors, there's quite a bit of coverage, 800 SNPs. In the immunoglobulin heavy chain, which is the main antibody chain, there are 5. This is a 1 million base pair region of the genome that is, I will tell you, enriched in an incredible number of SNPs and is completely underrepresented in that technology that all of us in this room used to do disease associations for a decade, meaning that we had a lot of an uphill battle to prove to the scientific community that there is a need here. To me, this is an inherent understanding. If your antibodies are fighting pathogens, then the variation in them should be associated with a response. You'd be surprised how many study sections didn't agree. So what we've done then is looked at the genomics of the locus itself. So I don't want to induce any PTSD for Immunology 101 class members in this room, but if you will all come back on a journey with me to remember that antibodies and T cells are encoded by the recombination, the DNA recombination of V, D, and J segments. Now, this is a cartoon from your standard Janeway immunology. The locus actually looks like this. All of those green boxes are V genes. So there's over 120 of them. The red boxes are pseudogenes, very similar to the V genes, but with point mutations that render them inactive. Diversity in navy, joining in yellow, and then those constant region, those tail genes down there in blue that we'll come back to. What this means, these gene blocks are each between 300 and 600 base pairs, so your standard short-read sequencing cannot tile through this highly repetitive region. Half of this is a segmental duplication in a way that can accurately reconstruct this. So when I explain the difference between short-read sequencing and long-read sequencing to a room of not sequencing experts, I explain it like a jigsaw puzzle. If you have a jigsaw puzzle of a sunset, beautiful purple, pink, blue, and it's 10,000 pieces, you're never going to know where that little purple piece goes. But if it's 100 pieces, then you get a lot of context. It allows you to stitch it together correctly, and that has allowed us to reconstruct these loci in a targeted fashion. So together with my partner, Corey Watson, we've developed both a DNA-based assay reconstructing those haplotypes of both the antibody-encoding genes and the T-cell receptor encoding genes, as well as a repertoire-based sequencing assay. This can be used for repetitive screening that captures the entirety of the molecule. And now that Clareo's launched, we've got product names. So this is GenotypAIR, AIR being adaptive immune repertoire. And then together we call the package of both assays CLAIRITY. And I want to tell you today how powerful these can be. So I'm going to start here by just proving to you that there's a reason for this. So again, taking you back to Immunology 101, you're taught we all have the same immune genes. We just mix and match them randomly, and that's why you're responsive to the flu vaccine and I'm not. This slide, this very complex slide that I won't go into detail, shows you that that is absolutely not true. So on the top of this slide, you see all of the V, D, and J genes along the top, and underneath you see the SVs, or structural variants, that are encoded in these locus. So this is a summary figure of 154 individuals from 5 major global subpopulations. What you see in the red dotted lines are an incredible number of large deletions that not only are different amongst people, each of these variants are reflected by at least 5 individuals in our cohort, but remove entire clusters of genes from these people's repertoires. So if you have a vaccine targeting one of those genes, but that gene is deleted in you, are you going to respond to that vaccine the way you're supposed to? Probably not. Not only are there deletions, there are insertions represented by those upside-down triangles. And then not only are there gene-based variation, but then there's allelic variation, or different flavors of these genes, right? So the gene segments encode the Vs that incorporate it into antibodies, but then those Vs can be altered by single residue changes that changes the allelic flavor and then also changes the function. And some of those genes, as you can see in the bar graph down on the right, have a lot of different alleles, 30, 50, 60, 100 alleles. So they must be selected for somehow and associated with disease. That was our bet. So just to show you, we are not all the same. And what we want to do at Clareo and with our work at the University of Louisville is harness this diversity to make medical treatments more effective for all, instead of ignoring that it exists and just assume and accept that they're always non-responders. The T-cell receptor is not immune to this kind of variation. This is a paper that we published a few years ago. Only 9 individuals, these are all 1,000 genomes individuals, very well characterized genomes. We used our targeted germline sequencing to sequence them, and in 9 individuals found 79 new alleles. So even though T-cell receptors are assumed to be more stable than B-cell receptors, we are showing here that they are highly diverse. This should be understood and explored further for understanding responses to immune therapy and other T-cell-based therapies. We're working now to expand this database. We've got over 300 individuals we're characterizing and hoping to publish soon in the largest T-cell germline database to date. So why does this matter? We know we're all different, but does it actually affect your response to disease? So the best example of this in the vaccine space is the current thrust in the HIV vaccine space of targeting very specific antibody genes to elicit neutralizing antibodies. And so one of the big targets at the moment is this gene V1-2. V1-2 comes in 10 allelic flavors. So here I'm showing you V1-2*02. If you carry that particular allele as a homozygote in your germline of IG, you express that gene in over 20% of your B cells circulating in your system. That B cell is ready. It is ready to see a vaccine immunogen. It is ready to respond, and it is ready to protect. And in fact, in phase 1 and phase 2 trials of this vaccine in both Europe and Southern Africa, this vaccine's been incredibly protective in HIV exposure. However, if you carry a star O5 allele, you cannot find these B cells. They're incredibly rare. They're sub 1%. And again, even though that we're taught that if you have a single B cell specific for an antigen in your big toe and you get a vaccine in your arm, somehow that cell is going to find its way up there and respond, we actually don't know empirically if that's true. And so the lack of correspondence, or actually the strong correspondence between genotype and presence, is very worrying in terms of responsiveness to these vaccines. There was an expansion of this study, more individuals, more genotypes. The genotypes here shown on the left, the different combinations of allele flavors of that V1-2 gene. So you can see a nice distribution of alleles. And then they looked at how that allelic variation affected the presence of those B cells in the circulation of these individuals, and it was striking. This is one of the strongest dose response curves I've ever seen, and it's genotype-based. So the particular genotype you have here in the pluses, that's the star O5 allele, you have almost none of these antibodies in circulation, whereas if you have that star O2 responsive allele, they're very high. Now, the difference between a star O5 and a star O2 allele is 1 arginine to 1 tryptophan, so 1 residue change. And so is this just a frequency thing? Are we just looking at an EQTL here? No, we're not, because if you take the responsive allele and you change that tryptophan to an arginine and do surface plasmon resonance, that antibody now is no longer responsive to the immunogen. It cannot bind, it cannot act, it cannot activate an immune response. So not only do you have fewer cells to respond to your vaccine, but even if those cells do meet your immunogen, you may not respond. That is incredibly powerful. How are we going to create universal vaccines if we're not considering the variability of the people just in this room, right? Let alone the rest of the world. So we were curious. We have funding to do population immunogenomics. We've been sequencing thousands of individuals using HiFi sequencing, using our assays from 58 global subpopulations. What is the distribution of this allele? Can we predict how people will respond to this vaccine around the world just based on the genotypes they carry? And this is the data that we have. So and this is a bit of a complex stacked bar plot. There are 5 major global subpopulations across the top, broken into subpopulations along the bottom. So African, Native American, East Asian, European, Southeast Asian. The frequency of the allele is shown in the stacked bar plot. If you have a pink bar, you have star O2, good for you, you will respond. If you have anything other than a pink bar, you do not have a responsive allele. You will not respond as that vaccine intended. Look at the Southeast Asian bloc. More than 50% of those individuals have non-responsive alleles. That's not just striking, it's sobering. There are massive HIV endemic issues in Thailand, in East Asia, that we have one vaccine that has all hope, and it may not work in these individuals. And that's terrifying to me, actually, as someone who believes in the equitable application of medicine around the world. And when we showed these data to those vaccine developers, their response was, oh, that's sobering. Yeah, well, that's true. That's not quite the word I would have chosen, but at least they acknowledged that. So this really spurred us to think, okay, if this is happening here, where else is it happening? What other diseases could we use this technology to really understand? And so then we started thinking about how antibodies progress disease, how they actually affect function in the body, and we came back to that stem of that antibody, that constant region, which I will tell you is a fallacy of nomenclature. Dennis Burton in 1994 called it the constant region because it was more constant than variable. But I can tell you for sure that it is not constant at the nucleotide level. We just surveyed 105 individuals using our capture method, using the GenotypAIR tool. This is a bar graph looking at the alleles that we identified in those 210 haplotypes. We were able to completely reconstruct using HiFi data. Please note the second to the right bar. These are the genes that encode IgD, which is the receptor that we use during B cell development that no one actually really knows what it does. No allele that we identified had ever been seen in the database. The blue bar is what's known. The red bar is what we added with just 100 individuals. We increased the knowledge of genes and alleles in this locus by 235% just by surveying 100 people. Not only is it not constant, it is a black box. It is rare in the field of scientific research to feel like you are an explorer in a new land. And this is what this technology has enabled us to do, which was only able to be done because of the accuracy, the length, and the contiguity of HiFi data. If you look at where those novel alleles allocate based on population, not surprising. The more novel alleles are found in those less well-characterized populations, which I think, again, is a critical call for action to make sure that data representation is a major focus of all of our research in this room. So I want to pivot a bit away from the germline. You know, I am now the CEO of a company. It is bad business practice to tell people they need one assay and can go home. We want to talk about how we can use these technologies to survey individuals to understand how their antibodies are changing throughout a disease course, throughout aging. And so we developed a RNA-based assay to look at the circulating immune repertoire. This is a schematic of what the IgG transcript looks like, right? Those VDJ recombinant segments that are then spliced to constant region exons that form that tail of that antibody. Current solutions really only look, again, at that VDJ, that upfront area, or even more restricted at that hypervariable region at the intersection of VDJ called CDR3. What we've done is we've developed a long-read assay that grabs the entirety of the molecule. So again, here are other methods. The reason that they're that short is because they're restricted by NGS, not because there isn't information in the rest of the molecule, but because the sequencing can only go that far. We've developed a long-read method that can grab the entirety of the transcript and for the first time link variable antigen specificity information with functional variation in the constant region that changes how these antibodies work. And we do this, right, on the Revio. And we do it with Kinnex, which has been a game-changing technology development for us. We can sequence these at over the depth you can get on other short-read technologies at an order of magnitude higher accuracy. For immune repertoire sequencing, this is critically important. These receptors mutate regularly to adapt to the pathogen in their system. So if you see a variant, you need to be absolutely sure it's real and not sequencing error. That's what HiFi and Kinnex has allowed us to do. We've now expanded this to cover all of the 11 AIR loci, so that's all of the Ig receptors, IgG, M, D, E, A, light chain, heavy chain, TCR alpha, beta, gamma, delta. It gives us accurate VDJ allele discrimination and clonal lineage analysis you can do with any other repertoire technology, but then it also gives you variant detection in untranslated regions, resolution not only of the constant region, that tail of the antibody, but also subisotype resolution, which I'll show you why that's important in a second. And what we're really proud of as a company that has come up in the face of other immune repertoire profiling behemoths that will be not named in this room is that we provide transparent data and workflows. You don't just send us a sample and get an Excel sheet with no idea what happened in the middle. We will tell you about how we did the assays. We will give you the raw data, and we will share our workflows with you. So why does this matter? Why does it care? Why do we care? The first thing I want to show you is what the data actually look like. So this is an IgV alignment of an IgG4 molecule. Yes, the variable region on the far right, highly variable, a different color for every base. It's a mess. Don't look too hard. You'll get a migraine. But you can see in the constant region exons in gray that there are allele-defining SNPs. So that allows us from the RNA level to reconstruct haplotypes and get allele-level resolution of these antibodies. Why do we care? This is a Circos plot. It shows the alleles on the top half and the gene usage on the bottom half. What I will have you note, IgG1 and IgG2 account for 90% of the circulating antibody in your system. IgG3 and 4 are sub 10%, sometimes only 1 to 3%. But they are also the antibody subisotype in which autoantibodies are enriched. If you currently do antibody profiling with other technologies, you will have to be digging through the needle in the haystack to figure out which of these VDJs associate with the autoantibody reactivity. With our profiler technology, you can go right in and say, these are the VDJs associated with IgG3. And we know that IgG3 is pathogenic, so we can ignore all the other noise. And that really allows you to target and do antibody discovery the way we think it should be done. So I want to demonstrate why we think this is going to be really powerful for biomarker discovery and companion diagnostic development using this example of autoimmune myasthenia gravis. So ACHMG is a horrible autoimmune disease. It targets neuromuscular junctions, creating problems in swallowing and breathing in the worst of cases. And we know from thorough mechanistic studies that it's mediated specifically by IgG1 and IgG3 antibodies. We also know that the pathogenic mechanisms are of 3 types. So either an antibody that binds, an autoantibody that binds and induces complement fixation and destruction of the neuromuscular junction, internalization of the receptor, or an actual functional block of ligand-receptor interactions. Specifically, this complement pathway is Fc-mediated. This is something only our technology can resolve, and it is something that drives clinical decisions. People who have AChR-MG are either put on a pan-B cell inhibitor, rituximab, or they're given a complement inhibitor. But if you have one of these other 2 mechanisms, internalization or functional block, that complement inhibitor isn't going to do anything. It's just going to cause a bunch of anti-immune side effects. So if we could use our technologies to predict which of these individuals are actually using a complement-mediated block, then maybe we could help guide some clinical decisions and have translational potential where we can identify variants associated with mechanism and then help make clinical decisions where you don't go through treatment exhaustion, trying all these different therapies until you find out what works. So what we did is we took a pilot cohort. This was funded by the Rare Disease Network. We looked at healthy donors and AChR-MG donors. We did DNA sequencing and RNA sequencing using our profiler and genotyper technologies. I do want to shout out Easton Ford here in the front row who did all of this work for his PhD thesis. This is a tile plot looking at the different alleles that we found just in the first 7 individuals. What I want to call out is look at the key on the right-hand side. Any sequence that has an FL, either at the front end or the back end, was a novel allele. So, and not only did we find novel alleles, but we found duplications. We found people expressing 4 alleles and 3 alleles when you really, right, should only express 2, mom and dad. And what we found then, there's also structural variation here that can affect what your repertoire is. These are the alleles that were found in the healthy donor versus the MG cohort. In this case, your purple bar is what was known and your blue bar is what was not known. So just again, every time we sequence an individual, we find novel alleles. Overall, especially in the disease group, over 60% in the healthy group and almost 80% of the alleles discovered in the disease group were novel, not known to science, and not understood how they may impact the development of this disease. So now we're looking at those constant regions. Because we know that's the functional effector part of the molecule, is there variation there that can explain the pathogenesis of the disease? And when Easton went digging into the literature, he identified both papers from the 90s as well as papers from 2 years ago, showing that there are certain alleles that carry deletions in their constant regions that accelerate complement fixation. So ding, 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 we know that's a mechanism. And we wanted to look at that in our cohort. And so there are 2 alleles we hunted for that we saw in our literature. The major allele or main allele has 4 hinge regions, and then the short allele, which has 3 hinge regions. We looked first in the healthy donor data we have, those 210 new haplotypes, and we see that there is population diversity of these alleles, which is important to know. This could also associate with which populations are more at risk for developing these diseases. And then we looked at the distribution of these alleles in our donors. Now, it's not a well-powered study, right? Only 48 individuals. We did not separate these individuals based on their pathogenic mechanism. That wasn't known at the time. But even with that sort of noisy experimental design, we do see an enrichment of the truncated alleles in the MG group. We also, as I mentioned, see these people that express 3 alleles, sometimes 2 short and 1 main, sometimes 2 main and 1 short. And we dug into the genomic data, we saw not only do they have a duplication IgG3, which if you have a higher expression, because you have 3 alleles of the pathogenic variant, that intuitively suggests some risk for disease, but also found that in 30% of individuals worldwide, we also have a duplication in IgG4. I won't talk about that today, but that's very interesting for chronic cancer therapy. So I'm going to leave you with a few final thoughts, and I am here to plug my own company. So if you guys are interested in Clareo, if you're interested in doing long-read immunogenomics, please come talk to me. I would love to chat more about it. I think what was critical for us, this is 10 years in the works, what I'm presenting here today in 20 minutes, highly accurate long reads were essential. We could not do this with any other technology or any other long-read technology. Immunogenomic variation and data representation matter. This is incredibly important in understanding why some people get sick when other people don't. Understanding the extent and impact of variation will be critical for precision medicine. Anything that you do to your body, whether it's a drug, a therapy, or an infection that stimulates an immune system to do the job for you, will be impacted by this variation. And Clareo Biosciences was founded to fill these gaps. We are offering custom tools for service. Like I said, we opened our laboratory in February. We already have 3 customers with over $500,000 worth of contracts. We're really excited about the enthusiasm we're hearing in the community, and we're looking forward to developing companion diagnostics for specifically for immunotherapy, which I don't have time to talk about today, but if you're interested, I'd be happy to chat. I want to make sure I acknowledge my team, myself, co-founders Corey Watson, William Lees. There's our QR code if you want to find out more. Of course, I'm here. And I also want to thank my collaborators in the MG study, my University of Louisville team, and our funding, of course. And with that, I'm happy to wrap up and answer any questions.